Okay.
Okay, great. Thanks, Phil. So can everybody see my web browser that says Introduction to Tidy Transcriptomics? Okay, yeah, Stefano's giving me the thumbs up. That's great, thanks. Okay, so everyone, welcome to our Tidy Transcriptomics workshop. Your instructors today are myself, Maria Doyle from Peter MacCallum Cancer Centre in Melbourne, Australia, and also Stefano Mangiola from Walter and Eliza Hall Institute, also from Melbourne, Australia. There's a little bit about us there. And actually, Stefano will share the link of this material. This is our workshop material. And Stefano will put the link of that in the chat, so you have that. And this is what we're going to be going through today. So I'm going to start off just explaining a bit about the workshop, and then we'll also have a hands-on demo section that you can follow along with some little exercises. Okay, so today's workshop is going to be on how to perform analysis of single-cell RNA sequencing data and some bulk RNA sequencing data following the tidy data, so the tidy data paradigm. Okay, and we're going to show you how you can do this using some new packages in CRAN and Bioconductor. So these packages include tidy Syrah, tidy single-cell experiment, tidy bulk, tidy heat map. And these are packages that have been developed by Stefano Mangiola, and who's presenting today. And they enable you to perform analysis of transcriptomic data using tidy data principles. And there's a new blog that's linked to in the material here that you can read a bit more about the tidy data, tidy transcriptomics manifesto. Okay, so for today, hopefully you have some familiarity with Tidyverse and a little bit of familiarity with bulk RNA sequencing and single cell. But if you don't, that's fine. You can just listen along. And the outline for today's workshop is we've split it into 2 parts. So all up, it's 3 hours. And the first part will be talking about bulk RNA sequencing. And the second part, which Stefano will take you through, will be on single cell RNA sequencing. And it's a mix of some hands-on demos where we'll be using the RStudio Cloud, and it's got some little exercises in there. And we also encourage Q&A. So if you've got any questions at any time, please feel free to put them in the chat. Stefano will be monitoring that while I'm speaking, and I'll monitor it while Stefano's speaking, and we'll stop at points to answer any questions you have. We've also got a little Menti quiz for you to do a couple of little challenges if you want to try them. Okay, so we hope at the end of the workshop today, you learn a bit about RNA, the steps involved with RNA sequencing analysis, and also how you can use these with some publicly available data sets and create some visualizations that will help you understand the results. And to do this using tidy data principles. Okay, so but just before we get started, we have a Google Doc linked to in the material here that I'm going to open. And Stefano, if you could put the link to this Google Doc in the chat again, that would be great. And so this just has the links that you'll need for today. So for one, we've got the link to the workshop material that I'm going through. In case you lose the link and want to come back, you can find it in the Google Doc. We've also got Menti, which we're using for a few of the challenges. And this is the code here to access the Menti. And then at the end of the workshop, we would really appreciate if you could take 1 or 2 minutes to fill out this really short feedback survey we have for the workshop, because we like to try and constantly improve it. Okay, so we have a couple of questions for you before we get started. So this is to get an idea of your background. So if you can go to Menti, we've got 3 questions here. So I'll show you now. If you go to Menti here and then fill in this code. So I think I need to share it through Menti for you. Hold on 1 second and I'll present. Okay, so a few of you have already filled it out. So if I can see if I can share this for you so you can see the results here. Okay, so if you go to www.menti.com, you can type in this code here. So you could do that in another tab on your browser or on your phone, because we'll use this same Menti and code for a couple of the challenges. And just to start off, we've got a question asking if you can rate these 3 questions on a scale of 1 to 5, 1 being no and 5 being yes or a lot. So for the first one, will you code along and try out the little challenges that we've got? So 1 would be no and 5 yes, and then maybes in the middle. And also, do you have experience with transcriptomic analysis? So again, 1 no, none at all, 2 to 3 a little bit, and then 5 a lot. Okay, and same question for Tidyverse. So this helps us to know your background. Okay, so we've had 11 people fill it out so far. That's great. Okay, so we can see that most of you are going to try out the exercises. That's great. And the majority of you have some experience with transcriptomic analyses, and quite a lot of you have experience with Tidyverse. So that's great. Okay, I will continue on for the moment then. Okay, so we had a little bit of transcriptomics background. So as most of you have some experience with transcriptomics, I'll just go through this very briefly, and feel free to come back. This material will be available ongoing after the workshop. So you can always come back to this web link here and read after today. Okay, so what is transcriptomics? What we're going to be studying today. So that is the set of all the RNA transcripts in a cell, and it provides a connection between the genome, the DNA in our genome, and the proteins in our proteome. Although we also have some transcripts that are non-coding as well. And why would we study transcriptomics? People like to study transcriptomics because you can study the genome, the DNA, but that can be quite stable, whereas the transcriptome will change in response to, for example, different drugs. And the proteome is another option that you could study to see what's happening in cells, but the proteome is harder to measure. You generally end up measuring less proteins than you would if you're measuring all the whole transcriptome. Because by measuring the transcriptome, we can measure changes in expression of thousands of coding and non-coding transcripts. So to see how thousands of genes are behaving. And a possible experimental design you might have is you might, particularly if you're working in pharmaceutical area, you might have a new drug you want to study the effects of to see what it does. So you might have some cells that you're going to treat with the drug. And then you can extract the mRNA transcripts to see which genes are being expressed. And then you would compare that to a control where you're not treating the drug with drug. And you can see which genes expression are changing. Okay, and then just very briefly, how do we study transcriptomics? So we take our transcripts from our cells, we extract the mRNA, we create what's called a sequencing library, and we put that through the sequencer. And from the sequencer, we get these, what we call short sequence reads. So generally about 50 to 150 nucleotides in length. And then what we need to do then is to see which genes they come from and which genes are then changing in response to our conditions that we're interested in. And there's different types of transcriptomic analyses we can do. So the most common by far would be differential expression analysis, where we're comparing, for example, the treated to untreated samples and seeing the differences in the expression of genes. There's other, you can also look at the cell type composition, which will show you a bit about today. So that's where you have, say, some samples, tissue samples, and you might want to see, well, what types of cells are present. For example, what types of immune cells are present. And that's something you can do with our bulk RNA sequencing. or with RNA sequencing samples as well. So we'll show you a bit about that today. And then there's other things, other types of transcriptomic analyses you can do, which we won't be covering in this workshop today. Okay, and the next part, we're just showing a pipeline, a common pipeline for how we analyze bulk RNA sequencing data. So we take our raw sequence reads from the sequencer, we map them to a reference genome. So for example, to human or to mouse, if you're studying using a model organism like mouse. So we see where the reads, which areas of the genome they're from. Then we count how many reads we have for each gene. And then we do some data exploration on it, for example, creating PCA plots and creating other visualizations to see how the quality of the data, to see if there's anything funny going on or if it looks good. And then we go ahead and do our testing for differential expression between our conditions of interest. And so in today's workshop, we're going to be showing you how you can do this once you've got your counting, how you can do the data exploration and the testing of differential expression using Stefano's package called TidyBulk. Okay, and then in part 2 of today's workshop, Stefano is going to take you through single cell sequencing. So he'll come back to this part here. Okay, and most of you say you're familiar with the Tidyverse, but just briefly for those of you who aren't. So the Tidyverse is a collection of packages that enable you to perform data analysis according to the tidy data paradigm, which was developed by Hadley Wickham. And it's a very, very popular way to analyze data. And the tidy data paradigm, it gives a standard way to organize data within a data set. So every variable is a column, every observation is a row, and you manipulate data. So you use functions that have easy to understand names. Okay, and most importantly, the data structure remains consistent. So we always know that it's going to have our variables in columns and observations in rows. So it can make your data analysis much more efficient and quick when you've always got it in this structured way. And so the Tidyverse is a collection of packages that can be used to tidy, manipulate, and visualize data. And we're going to use many of these Tidyverse functions in this workshop today. And these are functions such as filter, select, mutate for creating columns, pivot longer for reshaping, and ggplot, extremely popular package for visualizing data. Okay, so now we are going to get started with the hands-on part of today's workshop. So this is where you can follow along with us if you want. And most of you who filled out the Menti said you would do that, so that's brilliant. So the link to get to the RStudio cloud that has the material for today's workshop is in the chat. Is that right? Yes, I think it is. Yeah. Okay, great. So what you need to do is to click on that link. I think you have to create an RStudio cloud account if you don't already have one. And then you should see something like this, where it has a temporary copy in red, and you need to click on the link to save a permanent copy of the project, the Tidy Transcriptomics project. And that's so then you can play around with the material, and any changes you make will be saved in your copy of the project. Okay, and then if you want to, after the workshop, so the cloud is only available during the workshops because it's been kindly provided by our Pharma conference organizers.
Oh,
so after the workshop,
it's available. Oh. It's always available.
Oh, so can they continue to use this?
Yep.
Oh,
yep.
Oh, that's brilliant.
Yep.
Okay, thanks, Phil.
Yeah. It's as long as they will have access to the classroom from now on, forevermore. Basically, we don't have any plans to take it down or to kick anyone out. So.
Oh, that's great. Okay,
yeah.
I didn't realize that. Okay, so you can come back then after today and go and play around with the material in the RStudio cloud, or if you do want to install it on your own computer, there are instructions here for how to get the packages and the versions that we're using. Okay, or you're welcome to just view the material, which is this actual page that I'm on right now. Okay. Oh, someone's saying they didn't have the option to save a permanent copy, but it looks like they're in. That's okay then, as long as you can
Yeah, I think
run it. Is that?
I'll jump in real quick and just make sure that it kicks over to a permanent copy. Just give me, carry on, and then I'll
Okay.
let you know here in 2 seconds. Yep.
Great, thanks, Phil. So yeah, any questions or issues like that, just drop them in the chat and somebody in our group will help you out. Okay, great. Okay, so that was what is transcriptomics and what is tidy transcriptomics. So now we're going to get started with the hands-on part, which will be taking you through tidy transcriptomics. So I am going to move to my RStudio, my copy of the RStudio Cloud.
Checking that. Yeah, so it's set up as an assignment. So it should be kicking over as a permanent copy. If you go and click on the link to join the classroom, you'll see under the projects for you, it should say start assignment or start, I can't remember, but once you click that, it will generate a permanent copy within the classroom that will persist for you. So, okay, back to you.
Okay, great. Thanks, Bill. Okay, so let me see if I can start my copy. Okay, great. I'm going to just move up and clear off what was already done there. Okay, just get my windows organized. Okay, so hopefully you see this vignette. So when you're in the project, I'm down in the files pane here. And let's see, so it's our markdown document that I've got open is in the vignettes folder. It's this tidy transcriptomics markdown file here. So if you click on that, you should see this file that I've got up in the file editor pane. And I think we've got some objects from testing, so I'm just going to remove those. Okay, and if you want, you can click on this outline button at the top of the editor page so you can see the different sections we'll be working through. So if you get lost, you can hopefully navigate through this. So this is the material that's rendered into the workshop, into the web browser page that I was already going through. So we've actually gone through the first section here and we're down at what is tidy transcriptomics. So I'm just clicking to move to that. Okay, great. Okay. So tidy transcriptomics then is a suite of packages where Stefano is the key developer, key brains behind these packages. And they're tidy bulk, tidy summarized experiment, and tidy single cell experiment. And they enable you to analyze RNA sequencing data according to Tidyverse principles. Okay, so this image here is showing how the different packages connect Tidyverse and Bioconductor. So if you don't know, Bioconductor is the largest repository of R packages for biological data analysis. So really popular, really great repository. And Syrah is a particular R package toolkit for single cell RNA sequencing data. So Stefano will explain the single cell part in part 2. But for the bulk RNA sequencing part, so the tidy bulk package enables you to perform an RNA sequencing workflow using tidy data principles. And there's also the tidy summarized experiment package, which I'll show you in a moment. And that enables you to use the Bioconductor data format, which is called summarized experiment, but it enables you to use Tidyverse style methods and principles with that. So as I'll show you in a moment, you can instead of having to work with this summarized experiment in this way that's a bit more tricky to see what it contains, you can work with it according to this tabular or what's called a tibble in Tidyverse. So a table, Tidyverse table is tibble. Okay. Oh, been disconnected. Hold on a moment. Okay. Okay, great. Okay, so tidy bulk enables you to perform the RNA sequencing workflow, and tidy summarized experiment provides a connection between the bioconductor summarized experiment format and the Tidyverse tibble. Okay, so that's what I'll show you. And Stefano will take you through the single cell. And just to mention, there is a blog for tidy transcriptomics that you can get to. The link is in the material. And that speaks more about the background to tidy transcriptomics. And okay, now we're going to get started with loading our packages. Okay, so what I'm going to do here today is I'm going to be running these chunks. So with this little arrow here, and then I'm going to explain what's happening. And so you can feel free to run them as well and make sure that you get what we get and that you can try out this analysis. Okay, so here I'm loading the Tidyverse packages, an example data set, and these Tidyverse friendly packages as well. And we've also got some settings because we make a few plots. So these are some customizations for the plot. Okay, and right, so for the bulk RNA sequencing part today, we're going to work with this published RNA-seq data set from this paper here, Himes et al. And it's got 8 samples from human airway smooth muscle cells. So it's got 4 cell lines, and each cell line has a treated and an untreated sample. So a sample that was treated with dexamethasone, and then an untreated negative control. Okay, and the sample has undergone RNA sequencing, and we've got gene counts. So read counts for every gene. So here what I'm doing is I'm loading the airway data and I'm having a look at it. So now, here we have summarized experiment format. And actually, hold on a second. I'm just going to restart this because I think maybe Stefano was testing. So it's already got in the tidy summarized experiment package loaded, but I want to show you what it's actually doing. So I'm just going to restart my R and then run up to here, all the code above. Okay, and now I'm going to load in the airway RNA sequencing data. Okay, so this is what it looks like. The airway data set is in a bioconductor package. It uses the bioconductor way of representing transcriptomic data, which is this summarized experiment format. So if you can see, it's a bit tricky to see what exactly does this data look like. So a nice thing about the tidy summarized experiment package that Stefano's developed is it enables you to see the summarized experiment data set in a tidyverse way. So you can see now it's got columns and rows, so we can more easily see what it contains. And so this is telling us that this is a summarized experiment tibble abstraction. So it's still a summarized experiment format, but tidy summarized experiment enables you to view it like a tibble. Okay, so here we can see now that we've got genes, so gene identifiers in our first column. So these are using these ensemble gene identifiers. Then we've got our sample IDs in our second column. And then we've got how many counts we have for each gene in each sample. So this is our expression level of the gene. And then we've also got some information on which cell line the sample's from. And then the DEX column tells us whether the sample is the treated one or the untreated one for the cell line. Okay, so tidy summarized experiment enables us to view summarized experiment format in a tibble way, but we can still use summarized experiment commands. The summarized experiment has its own functions and commands such as assays, and they still work. Okay, so this is Next part is just some examples of how we can use tidyverse commands, such as filter, select, group by, summarize, and mutate to explore this summarized experiment object thanks to the tidy summarized experiment package. So for example, if we want to choose the rows for just our treated sample, so we've got 4 treated samples, we can use the standard tidyverse filter command. So that's saying for the DEX column, just give us the treated samples. So you can see now we've got 4 samples here, before we had 8. Okay, we can also use the tidyverse select command, which enables us to choose columns. So for example, we can select our sample identifier column, the cell line column, and the DEX column. So we can choose those there. And we can combine tidyverse commands such as group by and summarize if we want to, for example, calculate how many counts do we have for each sample. So we want to know our total read counts. And this enables us to see that for our 8 samples, how many counts we've got, around 20 million on average for each one, which is pretty standard for bulk RNA sequencing. And if we want to create any columns for our data set, we can use mutate. So the standard tidyverse mutate command. So for example, we can create a new sample name column that has shorter sample names. So we can remove this. They've all got this SRR1039 prefix. So we can remove that bit and just keep the unique part of the sample identifier. And that will make it nicer, smaller to fit in some of the plots we're going to make. Okay, so here we're using standard tidyverse mutate with tidyverse string remove. And then selecting the columns just to show the sample identifier column before and after. Okay, so that's using tidy summarized experiment, which enables us to work with summarized experiment objects in a tidy way. Now we're going to be moving on to actually analyzing bulk RNA sequencing data, which uses the tidybulk package. And tidybulk, developed by Stefano, has many functions. So they're shown here that enable you to do different things with the RNA sequencing data. And we'll see some of these in this workshop, but this is to let you know there are some others there as well if you want to explore them in your own time. Okay, so now we are going to set up and prepare our data for RNA sequencing analysis. So we are going to create a column with the shorter sample names, just as we did above using mutate. So to make a column that just has the unique identifier. And we're also going to create, so use mutate to create a column with the gene symbols. Because at the moment, we've only got those gene identifiers, the ENSG, but often we want to work with the actual gene names. So whether it's like BRCA or TP53. So we can do that by using other bioconductor packages to get the gene symbol. And if we take a look at the output, what we can see is we have our new sample name column here as before, and also we've got another new column added with the gene symbol. Okay, so now we move into the RNA-seq processing. So one of the things we do in RNA sequencing analysis is to filter out, remove genes that don't have much expression. So we do that because it can reduce our power to detect differentially expressed genes, and they don't have much information first. They might only have 1 read or 2 reads per gene per sample. Okay, so we can do that using the tidybulk keep abundant function, which uses edgeR under the hood to do the filtering. So we just need to say which column has our groups. So in this case, it's the DEX. Okay, and after running that function for filtering, we should see we get a column called .abundant, which has true for the genes that we filtered, which in this case, yeah, we have filtered. Okay, and the other step we perform in RNA sequencing analysis is to scale our counts across the samples. So this is also called normalization. So we want to normalize for technical differences between the samples. I'm just going to pause for a moment because I see a question has come through. So from Anya, why do some column names start with dot? So that was something Stefano, what was the reason for that again?
Yes, so the convention here, this is true. Well, for this data set, the feature and sample columns basically are not explicitly named in the summarized experiment, as are column names and row names, while here we give them names. So the dot is to indicate that those column names are given by us because are absent in the, in the rectangular matrix that is proper of the summarized experiment that is underneath. While all the other columns are either existing column names for the metadata for the samples or existing column names for the metadata for the transcripts.
Okay, thanks, Stefano. Okay, so yes, we're normalizing for technical differences between the samples. So that can be if they have been sequenced to different depths. So for example, if we've got 40 million for one sample and 20 million for the other, we need to take that into account when we're going to compare levels of the genes. And there's more information on normalization in the link here. But TidyBulk has this nice function, scale abundance, that lets us do this scaling or normalization quite easily across samples and using a function name that's fairly explicit and hopefully understandable in what it's doing. So after this, then we should see that we have a new column called counts scaled. So this now has the counts adjusted for these technical differences between the samples. Okay. And I'm going to run this here to say, because this is all thanks to TidyBulk and Tidy Summarized Experiment, all using Tidy data principles, we can very easily create plots. So using ggplot and also use Tidyverse's powerful reshaping, so pivot longer, to, in just a few lines of code, create quite informative plots. So here what we're doing is taking that scale counts table here, reshaping it so that we've got a column that contains our counts and scale counts, so our raw counts and scale counts, and then plotting it, creating density plots, ggplot, so we can compare our distributions of counts for each of our 8 samples before and after scaling. So here we should see, or we hope to see, that the distributions after scaling are more similar to each other, which they are here. None of the samples look weird. So it's nice to be able to just interact with the Tidyverse ecosystem like that. And just as a comparison then between this TidyBulk, Tidy style analysis of RNA sequencing data. So this is showing you that with a couple of lines of code, we can filter our lowly expressed genes, normalize our counts, normalize our samples, and create our density plots. A few lines of code compared to the more traditional way of doing it using base R, for example, with EdgeR, where this code block is showing we'd have to type a lot more code to be able to create similar density plots. So there's more chance for typos or bugs, and it would take longer to do that. So TidyBulk helps you to analyze your RNA sequencing data more efficiently. Okay, so once we've done our filtering and our scaling, we then want to create some principal component plots or multi-dimensional analysis plots to see how our samples look. So this is a really key plot that we make when we analyze RNA sequencing data, because this helps us to see, do we have any outliers in our samples? Are our samples from our different groups, from our, for example, treated and untreated grouping together? And so this is one that we always do for RNA sequencing. So TidyBulk has a function called reduce dimensions, where we can give it our counts and we can choose whether to do a PCA or an MDS plot and or calculations. And so here I'm doing PCA, and by default, it will calculate the first two principal components and tell me the fraction of variance attributed to those principal components. So now we have a little challenge for you. So in Menti, where we want you to try and see what is the fraction of variance explained by PC3. So this is PC1, PC2. So what is that for PC? What is this fraction of variance for PC3? Okay, so it's in Menti. Can we see who the leader is? Okay, so there's supposed to be a little leaderboard with it, but doesn't seem to want to work today.
Later.
Yeah, we will get that working then. Okay, so I will show you the result. So you should see that. I'm just going to pop this down here. Oh, count scales. Okay, so there should be a, dims argument. So dims is .dims is 3. So you can change to how many dimensions. Reduce dimensions shows you, and you should see, did I get the output? Yeah, here. So it was 0.16. So well done, those of you got that. Okay, so what happens after we're running reduced dimensions is that it adds columns with the principal components to the table that we've got. So we get these columns for PC1 and PC2. And this has added them to the table that we had. So it's quite big. So it's 127,000 rows with 21 columns. And the reason it's so many rows is because it's one row for every transcript in every sample. But if we just want to know what are the principal components for the 8 samples we've got, we can use Tidybulks. It's got this pivot sample function. So here it's just the sample specific information. And with that, we can see what the PC1 and PC2 values are for the 8 samples. Okay, and once we've got the dimensions calculated, we can then use, again, Tidyverse ggplot to make a nice plot, a PCA plot for our data. So what I'm doing here is we've got the PC1 and PC2, and we've got each of our 8 samples colored by whether they're treated or untreated, and we've got shapes for which cell line they're from. So using ggplot's nice color and shape features. And we can also take advantage of Tidyverse friendly packages such as ggRepel to add the sample labels so they don't overlap. And so with this plot, we can see our samples are grouping nicely by treated and untreated, but also we've got one cell line that's a bit different to the others. So the square cell line here is a bit different. Okay. An alternative to or complement to principal component analysis is hierarchical clustering. So we can use that to see how our samples group or the relationship between the samples. So with Tidybulk has a function called keep variable that enables us to select the 500 most variable genes, because we can't plot them all. And with that, we can use Tidy heat map then to nicely create a heat map for this data. And we can add color categories along the top for whichever variables, columns we want to highlight. So here we've got our 8 samples, and we can see we've got our color gray for treated, red for untreated. So we can see they cluster nicely by the treatment. And again, though, we've got one cell line that's a bit different to the others. So these are the colors for our cell lines here. Okay, and for comparison again, so if we were to do this using base R, so not to do tidy style analysis, this would be an example workflow for that. So with this, you can see that the functions are a bit more difficult to understand. So TidyBulk is providing this nice, easy to understand vocabulary, scale abundance, keep abundant. And so, and it also, with the TidyBulk, tidy workflow, it requires a lot less creating intermediary variables because we are taking advantage of the Tidyverse pipe. So piping commands into each other, which makes the code nicer, less typing and less bug prone. Okay, so now we've checked the quality of the data. It looks pretty good. And what we can do then is proceed to testing for differences in expression between our treated and untreated samples. So TidyBulk is nice because it provides a wrapper for the most common RNA sequencing testing algorithms. So there's popular algorithms that some of you may have heard of, such as EdgeR, DEC2, and LimaVoom. And these are all really good methods for testing for differential expression. And TidyBulk enables you to choose. So you could choose to use one of them, or you could choose to use multiple and compare the results, which is a nice thing. So here we are going to use TidyBulk's test differential abundance method. And with this, we give it our counts table. We give it a formula. So we say which column contains our grouping information, which has the information on which samples we want to compare. We can add in additional variables. So we know that the cell line is contributing some variance. So we can include the information on that. So which cell line the samples are from. And then we can choose which method we want to test with. So here we're testing with the EdgeR quasi-likelihood method, and also the EdgeR likelihood ratio, also LimaVoom and DEC2. And this is adding a prefix to the output columns, so we know which output columns come from which method. And again, because this all works nicely with Tidyverse, we can use the Tidyverse pipe to simply combine the outputs from the different methods that we can then go and compare. Okay, and so we should see when we run that, we get our table. So we've got our same input table now with all these extra columns added. So from the methods, so from the EdgeR, Voom, and DEC2 methods. And the outputs that we're getting from those methods are columns with the log fold change. So how much the expression has changed for the treated versus untreated groups. And also a false discovery rate, so FDR, and p-values for each gene that's being tested in each sample. Okay, and our log fold change is log 2. Okay, and then we can use nice Tidyverse style methods to select and compare the outputs. So here, similar to the pivot transcript method I showed above, TidyBulk has a pivot sample method I showed above. TidyBulk has a pivot transcript method to enable us to select just the transcript specific information. And with that, we can select the p-value columns for each of the 4 methods. And we can use one of the Tidyverse friendly packages called gggali, has this nice method called ggpairs that enables us to do pairwise comparisons for the methods that we run. So here we can see that some of the methods are more similar to each other. So comparing the p-values. So the 2 EdgeR methods, as you might expect, are the most similar. And then the Voom and DEC2 methods are more different to each other. But they're a bit more different. They're still pretty good. Okay, and now we have another little challenge for those of you who want to try it. So that is, of these 4 methods, which method detects the largest number of differentially abundant transcripts? So using the p-value that's adjusted for multiple testing, less than 0.05. So depending on the method, the column is called FDR, adjusted p-value, or PADJ. Okay, so this will be a Menti question again. So we'll give you a couple of minutes to have a go at that. And I'll see if I can get it up on Menti for you. Okay, and again, this is an opportunity while we pause. If you've got any questions you want to ask us, please feel free to put them in the chat.
I think there was one question.
Okay.
If you see that.
Yeah. So for DSEQ2, somehow specify that I want to use truncated LFC. Well, so the ellipse arguments, the 3 dots, plus whatever arguments to the DSEQ2 fit function. I don't know specifically how the LFC is requested in a way to DSEQ2 from which function. I'm a bit more experienced in a jar, I have to say. But let me see 1 second while we wait for the quiz. So the answer is yes, if it's part of the fit function. And probably no otherwise, but these backend frameworks can be made flexible, of course, as we have done for HR. So for HR, there are many different flavors, and we accommodate them all with the specific name of the workflow, of the HR workflow that is underneath, that are all documented. So in case it's not, feel free to open an issue, and we will go from there.
Okay, so the winner for that challenge was Anne. Well done, Anne. Wow, you got that very, very quick, I think, because the quicker you get it, the more points you get. So well done. Okay, so there's a few ways that you could get that answer. So you could use Tidyverse filter to filter the output table for each column belonging to each method and then count. Or another way to do it is using Tidyverse's pivot longer method. So I'm just going to pop that one in here so you can see, just in the interest of time, I'm copying and pasting. Oh, actually, that one needs, that should be Dex. Or maybe, actually, okay, there's a typo in that method. I will fix that and get that. But in the interest of time, I'll move on. So we can go through the part 1, and then you'll have more time for part 2. Single cell. Okay, so that above is showing you how you can run multiple methods if you want. You can also just choose to use your favorite differential testing method. So by default, Tidybook will use the EDGAR quasi-likelihood method. So that's what it's doing here. And that will give you then just one column for your log file change and one for your p-value from that method. Okay, and again, for comparison with a base R non-Tidyverse method, this would be how you could do that similar style analysis. But again, just note the functions are more difficult to understand names, like less easy vocabulary. And also, again, you have to create a lot more variables whereas using the pipe enables you, the Tidyverse pipe enables you to use less variables, and so hopefully have less bug prone and quicker to type code. Okay, so now we've got our differential expression results. We can go on and visualize it. So here, what I'm doing is creating a volcano plot. So this would be a pretty typical plot to create for RNA sequencing results. So what it is it's showing us the log fold change. So the magnitude of expression difference on the x-axis. And we've got the significance of the gene, genes changing on the Y. So every dot here is representing a gene. So the genes that are more up-regulated, being in the treated samples compared to the untreated, are on the right-hand side, and the genes that are down-regulated in the treated are on the left. So that's a basic volcano plot using ggplot. But something to note is that Tidybulk package allows us it takes advantage of being able to create custom scales for ggplot. So it enables us to plot on the y-axis a log 10 reverse, because normally we plot for volcano a negative log 10, and it can make what the actual p-value was difficult to see from the plot. But Tidybulk enables you to plot the data and represent it as the actual p-value for the gene, which is nice. Okay, and just to show, then we can take advantage of the extensive customization abilities of ggplot and Tidyverse if we want to create more informative volcano plots, such as, for example, labeling our topmost expressed genes, which is what I'm doing here. So I'm taking the genes, identifying the genes with the smallest p-value, and then I can go on and use Tidyverse's mutate to say how I want to color and customize the plot and create a nice looking volcano plot like this, with the most significant genes highlighted in red, or sorry, the most upregulated and downregulated highlighted in red, and our top most significant by p-value genes labeled. Okay. And a nice feature or nice function that Tidybulk has, or a really cool function, is that it keeps track of the methods that you're using. So the methods such as EdgeR that Tidybulk is using under the hood. So it's got this function called get bibliography, that if you give it your object, your R object that you've been processing with Tidybulk, it will give you all the references that you are associated with your analysis. So you could then, this is in bibtext format, you could then pop that into your reference manager and have your references already there for your report or your paper or whatever. So that's really nice. Okay, so that is the differential expression analysis. So for comparing treated and untreated samples and seeing which genes, the expression of which genes are changing. So now I'm just going to briefly show you how you could perform a cell type composition analysis using Tidybulk. So we're going to use a different data set here because this is more for if you have tissue samples as opposed to cell lines that we've been using. And if you have tissue samples and you want to know what cell types are present, and if there's differences in expression between the cell types, then Tidybulk has this deconvolve cellularity function that can enable you to do that. So for this part, we're going to use a subset of the breast cancer data set from the cancer genome analysis. So it's a big publicly available data set. And we're going to load in that example data set here. So here, this has got 22 samples. So from these breast cancer patients, and it's got information on, for each of these samples, information on genes and what level they're expressed at, similar to before. And what we're going to do then is to use Tidybulk, deconvolve cellularity, and to identify what cell types are present in these samples based on their gene expression levels. And so TidyBulk here is using a method under the hood called CyberSort. So it's a very popular method for doing this type of deconvolution analysis. But similar to like what we saw with the test differential expression, TidyBulk also has options for running a couple of other methods if you want to try those and compare. And CyberSort identifies what cell types are present using the signature called LM22 that is used to define 22 immune cell types, 22 human immune cell types. So it's going to try and identify these in the sample. And when we run that, we get a table added, or columns added to our table that tells us what proportion of these cell types TidyBulk or CyberSort is detecting in each sample. So for B cells and monocytes, macrophages, etc. And what we can do then is we can use Tidyverse methods, so pivot longer, to reshape this data that we can then go and plot with ggplot. So we can create one column that gives us the proportions of each of these cell types in each of our samples. And we can then use ggplot to create a nice bar plot that gives us, for each of our samples here on the x-axis, we've got what proportion of these 22 cell types, 22 immune cell types are in each sample. So we can see our macrophages, mast, NK cells, etc. in a nice visual way. So we've got one final challenge for part 1 here, which is what is the most abundant cell type overall in these breast cancer samples? So which cell type is present the most across these samples? Okay, so it's another Menti question. So let's see if I can get that question up for you. Okay, and again, we'll give you a couple of minutes, a few minutes to have a go at that. And again, if anybody has questions that you want to ask about what we're showing you now here, please feel free to go ahead in the chat and ask us. Oh. There we go. What is the most abundant cell type overall? Was macrophages, M0. So well done. Most of you got that one. And for our little leaderboard. Ooh, it's a close one. Okay, Anne, actually, okay, Anne, you're doing very well. Okay, well done. And let's move back to RStudio Cloud, and I'll give you again, in the interest of time, I'm just going to pop the solution in here. So what you can do is take the data set we have, you can group it by the cell type, you can use summarize, Tidyverse summarize to calculate the median for each proportion, for each cell type, and then use Tidyverse arrange to sort descending. So you should see then that macrophages have the highest proportion across the data set. Okay, that's great. And so we've come to the end of part 1, so the bulk RNA sequencing analysis. So hopefully you've seen in this part how you can represent RNA sequencing analysis in a tidy way. So if you've got a summarized experiment format from Bioconductor, you can represent it using tidy summarized experiment, and you can also analyze it. So you can use Tidyverse's filter, mutate, group by, summarize to have a look at the data. And then to actually perform an RNA sequencing analysis, you can use the TidyBulk package, which enables you to take advantage of the Tidyverse with transcriptomic data and to analyze it and visualize it in nice ways. Okay, and some of the key steps in a bulk RNA sequencing analysis are filtering transcripts that have little expression, scaling counts to adjust for differences in sequencing depth and composition, and then testing for differential expression then. And there's lots of ways you can visualize the data. And so we've shown you some of the more common ways of visualizing RNA sequencing data and how you can do it in a tidy way with Tidybulk and its interaction with the Tidyverse. And so Tidybulk is a really nice package that enables you to perform multi-method analyses. So you can use methods like very popular methods such as DESeq2, EdgeR, LimaZoom, and also analyze cell type composition using methods such as CyberSort within Tidybulk. Okay, so that brings us to the end of part 1. And if you have any questions on bulk RNA sequencing analysis, please feel free to ask them now in the chat. No.
BRCA set up the output from the convolved cellularity. Seems the result is not very memory efficient. It will create a problem when analyze large-scale data sets. Well, which output was the long format of the cell types? Well, Maria, can you print the BRCA cell type variable?
What was that? The
BRCA cell type variable. So
BRCA
in brief, the
cell type, the long one or the pre-long?
Okay, so this one is memory efficient because, as you can see in the header, is a summarized experiment table abstraction. So what we are abstracting is a simple summarized experiment. So as summarized experiment is quite efficient, as is organizing information need, we are just exposing that information to you in the form of a data frame. Okay, this is not actually a data frame. You can visualize it as a data frame, you can manipulate it, you can plot it and so on as a data frame, but the information is stored in an efficient way because it's a summarized experiment abstraction. So yes, you can go to pretty big data set as you would do with a summarized experiment. Nothing is changing basically here. And then is there a paper reference to show the sensitivity of differential expression by comparing this to EdgeR? The answer is most surely yes, although I don't know in the top of my head. Yeah, these are very, how can I say, popular and methods that have been developed years ago. So the answer, I don't know. I don't know if Maria, you know, on some of top of your heads, but there must be a review and benchmark that compare those two. Definitely, the answer is yes. Just a matter of looking for them.
Yeah, there's been a number of papers, I think, that do comparisons. I think it can depend on the data set, the number of samples. Let me see. There's one here that I'm going to post in the chat.
Well, I published one regarding outliers. So how robust these methods are against outliers. Well, I just post this paper as well.
Okay, great. Because they're all good methods. I know LimaZoom is good if you've got hundreds of samples. It's a lot faster than EdgeR and DESeq2. So if you're doing any of these big analyses, it's a good one.
There is the implementation of robust implementation of likelihood ratio of EdgeR that outperforms all the common methods out there if outliers are present. So it's very robust against outliers.
Okay, and if no other questions at this point, I'll pass over to Stefano now. And again, if you have any other questions, just keep popping them in the chat as they come to you. Okay, I'm going to stop sharing now.
Right. I will, let me see. Do you see my screen?
Yes.
Big enough? Are the fonts? I always put very small fonts. Is the Zoom enough for the users?
That looks good.
Alrighty. Okay, so let's go to the second part. We have shown as we can work with bulk RNA sequencing data, providing a streamlined workflow for analysis and also a tidy representation. So these two packages were tidy summarized experiment for data representation and manipulation, and tidy bulk as a tidy workflow for data analysis. Now, we will talk to you about the software we have developed for a single cell data representation and manipulation. Just a quick reminder, actually, in the meanwhile, can I get into the chat how many of you never did single cell analysis, single cell RNA sequencing analysis? And maybe Maria can let me know how many people of the 20 participants we have. All right, so just a brief introduction. Single cell sequencing data adds another dimension to the data. So in this case, we can recognize which molecule of mRNA that we sequence is coming from which cell. And so cells become a collection, all right, few of us, become a collection of transcripts. And so we have the 3-dimension are how many the transcripts we have, the cells that we have, and if we have a multi-sample analysis, as is common these days, we have also sample information. And so you can imagine you can have a much higher resolution of your data. You can start to do a much more complex and informative analysis. And so this again is a very standard analysis for single cell data, and is quite more complex than bulk already, but you can do many more things as the field is developing year by year. And so we can, get our reads, map them to the genome. We can get our transcript counts, and we can go to data exploration. That's where a lot of the data curation and manipulation happened. And so our packages, tidy single cell experiment and tidy Surat, facilitate these operations with a tidy paradigm. And then we can go to cell clustering, classification, so on and so forth, trajectory analysis, cell-cell communication analysis, and so on. So it's quite more richer. Here, the goal is not to show you how to do single cell data analysis, but to show you how you can do many of the steps through this analysis with a tidy paradigm using our packages. For the people here that they do not have so much experience on single cell, obviously it is not 1.5 hours is not enough to teach you that, but you can get a taste and you can do your learning, come back to this workshop. This is will be active here, but we have plenty of resources online also about this workshop, and you can come back to here to learn more. Okay, so we start to load all the libraries as before. We use a few of them from the Bioconductor repository. The two bigger analysis framework for single cell data are included in Bioconductor, where the community contributes with software, and as well there is Surat, is very popular from the Satya Lab, where that is more monolithic. We will show you that as well here. The idea is that, again, we provide packages for data representation and manipulation with a tidy paradigm, and we go toward harmonization of these two big frameworks. So the data will look the same irrespective if they are single cell experiment or Surat, and we will be able to manipulate the data in the same way using the tidyverse vocabulary that we might already know. All right, so we import a PBMC dataset for this exercise. Again, we start with a single cell experiment object from the bioconductor, and you can see that this is a little description of the dataset if you want to know more. The bottom line here is that the single cell experiment again is a quite complex and hierarchical object, but when we print it on screen by default, we get a summary. It's pretty similar to summarized experiment Maria showed you before. Some dimension information, which assays, some feature names and cell names, and so on and so forth. Okay, what happened when we load tidy single cell experiment? As before, now by default, the information about cells are exposed to us, and we are able to visualize them here and also manipulate them directly. Also in this case, this object is presented to us as a single cell experiment table abstraction. This information includes cell metadata, but as well reduced dimension and all sorts of information cell related. If we choose, we can use join transcripts, as I will show you later, to join transcript information in this representation as well, to do calculations, visualization, and so on. As before, this object is still a single cell experiment. So we can, whatever workflow we were applying before that we are used to, we do now. Nothing has changed. If we call a single cell experiment a function assay names, it works perfectly fine with our tidy representation. So nothing is taken away. We are just creating the link to the huge tidyverse ecosystem that now you can also use if you choose so. All right, so I will go through, as Maria showed you before, some of the simple commands that you can use to manipulate this object. As you can imagine, the power of tidyverse is not so much the single methods by themselves, but by the fact that you can build a full workflow with them, and their integration is really what to give power and efficiency. So I will just show a few of them. In this case, we can filter our single cell data set where the identity is equal G1. It's pretty easy. These are the cells that remain to us for further analysis, and a valid single cell experiment object is returned to us. We can select some columns that are cell-wise. In this case, we just select cell and some statistics. This is still a single cell experiment object. In this case, we select, we omit key columns and is not a valid single cell experiment object anymore. We get in return a simple table. The package will tell us so. And with that table, we can do whatever we want with it, further analysis, visualization, and so on. In this case, we combine two of the operators, mutate and select. We can let's say, clean up our data set, putting lowercase on an annotation column. And for you, I select two of those columns. As a cell ID column is missing, this is not a valid single cell experiment anymore. So we just get a table in return. Similarly to what we would do with data frames, with tidyverse, we can merge single cell experiment objects, simply use a bind rows. So you can imagine as two data sets are bind one on top of the other. In this case is a quite trivial example, as we are binding a data set to itself, but you can imagine a real application for this, as we have we want to integrate multiple data sets. In this case, we get a message that duplicated cell IDs have been made unique. All right, so here an example that you might find in this data set. We have a file column where when we imported our data set, we use some multi-sample files to import, and we have tracked the file path. Now, this file path can include some information that we might want to retrieve. We can do simply that with some TidyR quite powerful expression, which is extract. In this case, we are using a regular expression to extract the sample ID, the data set ID, and the groups from this path using, again, a regular expression. This is just an example, but if you know or learn the Societyverse grammar, you know how powerful and easy it is to make manipulation. And now you can do it directly on your single cell data. And this is in return, basically. In this case, samples is the data set plus the group for the sake of this workshop. All right, so for the next part of the workshop, I will just present you a case study, very simple, when I will go through some of the common steps for doing single cell data analysis. And I will show you as we can integrate both bioconductor libraries as well as Societyverse style programming in a very effective way. Let's say one of the first things we want to do with single cell data is quality control. So some of the droplets we get out of the machines and the analysis might include cells, but might be empty, might include just ambient RNA, or might include cells that are dying. So it's important to just select the droplets for single cell that includes healthy cells. In this case, we apply a quite common workflow for bioconductor. We use the raw names of our data, which is our transcript IDs, and we get the location through this database. Okay, it's warning that 10% of our transcript didn't have a location. That's completely fine. Normally, the quality control is done. This is a multi-sample data set. Quality control is done per sample. This workshop is quite relatively short. So we will just show you a case for a sample, and then you can imagine as you could iterate those for multiple samples. I will mention that as for who knows nest and map functionality. Actually, I'm curious again if you can put in the chat if you know and or used ever the nest and map paradigm for Societyverse. And I will mention how you can do this quality control per sample in a very programmatic way. Anyway, so let's go with one sample. We can use the filter, again, functionality to just filter one of those. Yes, I've done this already. And now we can see that one data set includes just 300 cells. And now we use per cell Q metrics, which is a utility from bioconductor. And again, we want to do this operation for the mitochondrial gene. So we are trying to derive statistics about the mitochondrial content in our cells. And we use the location that we have detected before, as you can see, where the location is equal to MT, so mitochondrial. And these will create a data frame where as variables, there will be some statistics for mitochondrial presence here. We can see some self-explanatory as subset mito%, where we have the percentage of the transcript output for a cell that is belongs to mitochondrial. We can do, now this is a data frame object. And for converting into a object compatible with tidyverse, we can simply call as table, where we define the raw name as cells, and table is simply a table that has particular properties created by the tidyverse community. And now, because this is a table, we can use mutate, and we define this cell has high mitochondrion according to a condition, and this condition is given by this function that is called isOutlier. We want to type equal higher, so higher mitochondrial is our outlier, and we want to base the statistics on the percent of mitochondrial transcription. So this, what is doing is basically just calculating which are the cells that are very high mitochondrial, that have very high mitochondrial transcription, and label them as high mitochondrial. As you can see here, a new column is added to this table, which is called high mitochondrial, and is a Boolean, either false or true. And now one pretty neat thing that we can, with the tidyverse grammar, integrate information to our single cell dataset. What is the information? Where we just calculated statistics per each cell, you can see here, of the mitochondrial transcription, and we have just labeled the cells that we want to discard, which is high mitochondrial equals true. So what we can do is to just use left join again, as if we were interacting with a table. We left join this information by cells. As you can see here, what we get back is our single cell experiment object as abstract as table, but now all information about mitochondrial have been added here. And am I filtering here? Okay, I see. I'm plotting before. Well, now you can imagine you can do this procedure for each sample. And we have already prepared a data set for you, just for simplicity and for the sake of time. And now we get a print out here. As you can see here, we get our 3000 cells back, as we imagine, with all our information for mitochondria. Okay, the next block is how usually after we calculate the statistics, we do some visualization to see the distribution of possibly dead cells. There are many ad hoc visualization from Bioconductor where we can visualize these properties. But for the sake of this tutorial, I will show you how we can reproduce the same visualization in a custom way with particular aesthetics that we might want to use for publication. And because this is the table abstraction, is very natural to interact to use a ggplot for this. And it basically comes for free with the use of tidy single cell experiment. One thing we can do for building a multifaceted plot with ggplot is the function pivot longer is very powerful. So we reshape the property we are interested in, and then we use those property to build multi-facet plots, so quite complex plots. So I show you just here when we pivot longer, these are three properties of our cells. One is how many detected transcript we have per cell. One is the total transcript output of each cell, and one is the percentage of that is from mitochondrial. As you can see, and okay, we are defaulting to default column names. You can see here that we have reshaped these three properties, and we have the value here. Now it's very easy to use the powerful ggplot to display these three information one next to another. As you can see here, we have our detected mitopercentance and total transcript outputs, and we've decided to use some custom aesthetics. We have customized the color for the outliers, We have customized the size for the outliers and as well the transparency. Okay, this is just an example, but it's as easy to use ad hoc visualization as it is to build custom ggplot in this case. And we can see that some cells for each sample, they have very high mitochondrial transcription, and these are likely dying cells. These cells have also a low transcript abundance overall. So this is also indication of dying cells. That's great. So we have an idea that the cells that are labeled as high mitochondrial are indeed probably cells that we want to discard. And what do? Simply filter the cells that are high mitochondrial equal false. In this case, we visualize this object. We have eliminated roughly 100 cells. Okay. All right, so another step, quite important step, is data scaling, as we have seen with bulk RNA as well, because different samples have been sequenced with different depths, but also because different cells have different amount of RNA. Some cells are bigger, some cells are smaller. Now, is not always the case. We want to scale by cell content, because that could be a biological indication, but nonetheless, sometimes we do. So in this case, we do exactly that. And also in a very essential thing when we do multi-sample analysis is data integration. So we want to avoid technical differences to be able to map cells in the same space, so to speak, to identify cell cluster and characterize them further. And so in this case, I will show you a combination of tidyverse commands with some bioconductor commands as well. I mentioned about the nesting that I'm not sure anybody has encountered before. I suggest to you to introduce it into your language. For tidyverse is one of the most powerful functional elements for functional programming in R. Anyway, I show you now that we can group our single cell data set arbitrarily. In this case, we group according with data set. As we can see here, we get out a table where you have data set in one column, and the other variable is actually a single cell experiment. And we can use, if you ever encounter it, we can use the map functionality to apply whatever process to each of those elements, or do other things, as I will show you now. Now, because we have grouped our sample, we can compensate for batch effects for each of these data sets independently. And so that's what we do, because we have separate them. And then after a batch correction, we gather this data as a list and send it to FastMNN for data integration. And because FastMNN strips a lot of information for some reason out of the cell metadata, we can simply left join the metadata that we had before. This is also very convenient. All this is done with pipes, so we don't need to create temporary variables or redeclare variables through the workflow. So this becomes also quite robust, as we have less chance of error. I show you simply here that the metadata we have added back to our data set is simply our data set. We transform it as a table. And again, we left join this table to our data set. And so the result is a data set that has the information as before, but also has some dimension, reduced dimension that are corrected. Okay, this is by default, the first 5 are displayed for us. And these are the dimensions where all cells have been integrated together in a common space. Feel free to ask questions. Here I'm aware for it's quite complex things, concepts for people that never had the pleasure to work with single cells, so don't be shy if you want something to be clarified. Okay, so we go to the next step. Usually after we integrate cells, so cells are now in the same space, we try to identify cell clusters. So in this case, we just use, this is nothing particularly tidy in a way about this code block, is simply some common bioconductor code where we update the metadata that we have with some cluster labels, and we use SNN graph for that. And I will show you now what information we have extracted. Here we called a select, which is from Tidyverse, and I will show you as we have created a new column called label that includes the clustering IDs of each of our cells. So these algorithms is a graph-based method as identifies cells that are similar to each other and label them as the same cluster. As you can see here, we can swap between bioconductor commands and Tidyverse commands seamlessly. I will show you the visualization of the cells in space in a little bit, just to show you that you can simply create summaries of your table very easily, as you would do with the actual table. Sorry, summaries of your data set as you would do with the table. In this case, we are counting the number of cells in each cluster. And you can see here that we have a quite heterogeneity. You can see the biggest cluster and so on. Okay, so in this case, again, after we have identified a cluster, we might want to visualize them in 2 or more dimensions. And we use a function from Skater called RunUmap. Maria showed you before the PCA. So the principal components are a lower dimensional representation of your 20,000 transcripts. So because you cannot visualize 20,000 dimensions, we try to describe the heterogeneity of the data set with just 2 dimensions. And so we run PCA. PCA is not enough for single cell data because it's very highly dimensional and because the cells here are kind of connected one another. And so there are cells that are similar with other cells, and there is a kind of a hierarchy. So a linear approach like PCA is not enough anymore. So there are other techniques such as UMAP and TSNE that are non-linear, and they learn actually this complex space that resolve better the cell neighborhood, but also in this case preserve the long distances. And you can read about that. It's obviously a pretty interesting and complex algorithm. So we identify this reduced dimension, this UMAP dimension. In this case, we default to 2. And we use our corrected reduced dimensions, meaning our integrated dimensions. So we go from a reduced dimensions to another reduced dimension, which is UMAP. As you can see here, simply our UMAP columns are added to our data set, and they are visualized in this tidy representation. Okay, so there is another challenge for you. It's quite simple, even though you haven't understood completely everything about the procedure so far. This is more a challenge that involves some summarization. So the question is the variability of the first UMAP dimension when we calculate 2 components equal more or less than when we calculate 3 components? Meaning, if we calculate 2 components or 3 components, is the first dimension always the same, or it depends on how many components we calculate? Okay, and again, go on Menti, and I don't have the admin on Menti, but Maria can assist here and put your answer there.
Yeah, I've started the Menti countdown. So we've already had one person reply. So yeah.
I give you a hint. Okay. So as you can imagine, you can run UMAP with 3 components because we want to know the difference between 2 and 3. And you can simply understand how much variability the first component is summarizing the data. You can imagine, you know, how to identify how variable a quantity is in a way. So it's the same thing. Grab the first dimension and understand which is the, you know, if it's more or less variable in the 2 cases. And we show you before how to summarize the data. And I can tell you it's a summarize function, as easy as that. And as well, you can ask question, that's the right channel. So. Yeah, so does the tidy single cell experiment works with sparse matrix and even HDF5 format in the backend? So the answer is yes, because the tidy single cell experiment, again, is not an object. Tidy single cell experiment is just an abstraction. So what is underneath is a simple single cell experiment. And usually simple cell experiments, they have sparse matrices because of efficiency. So we are simply creating an interface for single cell experiment to you. There is nothing different from that. And this is also true for summarize experiment. We are creating just an interface that you can interact with, but underneath there are, you know, the same object as you would expect. That's why you can use your workflow as before with no difference. That's why the efficiency remains the same because the object remains the same. That's the actual reason. And later on, I will show you what happened with the Surat objects. And I will show you how the transition from single cell experiment to Surat, when we use our tidy abstraction, is very easy. So basically you can do whatever I did so far with the exact same code. I mean, for what is data set manipulation with Surat, with no differences. How are we doing?
Had a few people fill it out. Do you want to just give another minute?
Maybe we can. Can everybody write in the chat whether they are if anybody is still trying? Please write in the chat. Otherwise, then we might go on. Okay, we might go on then, and I will show the answer.
Okay, I just ended it. And I'll just let you know. Oh. So most people guessed that it was less, but the correct answer was more. Is that right, Stefano?
Ability is less. Maybe there was
so maybe it was a typo in the question.
Yeah. So I think if if most people guessed as less, they are right. As well, I will tell you that UMAP differ from PCA as the components of PCA, the reduced dimensions are independent of how many dimensions you choose, while for UMAP, this is not the case. So in a sense, if you increase the number of dimensions, each dimension will describe a smaller variability, okay? And this is one possibility for finding the solution. Again, we propose an elegant solution, which is hard to approach in a sense if you have never programmed with Tidyverse, but the same could be done in a simpler way, going, you know, for the 2-dimension and 3-dimension separately. But what can we do here? We are left joining two data sets. One where we calculate the dimension 2 and one we calculate the dimension 3, and we convert them as stable. And then the UMAP one will be a duplicate name, will be .x and .y. We just summarize their standard deviations.
So the variability of the first UMAP dimension is more than, is it more?
The first UMAP, yes.
Yeah.
So maybe okay. That's correct.
Maybe,
in that
okay,
case.
so that person was, yeah. So more was correct.
Okay, yes. So as I explained, while you calculate more dimension, the variability decreases. So the answer. So who is the lucky one or the very smart one? You got right. Yeah, it's more in the case of 2-dimension than 3.
Okay, so well done, Anne, I think it was again.
Amazing, wow. And okay, that's it. And one other nice thing that I want to show you is that we can interface ggplot with our single cell data now, but we can also interface with Plotly. Is a very powerful library. Again, because of the study representation. And so since we have talked about 3 UMAP dimensions, you know, if we want, we cannot stop to 2 dimensions that we usually use, but we can provide to Plotly 3-dimension as they are part of our data set and visualize a interactive 3D plot. Maybe I should have executed this, where we can color by our clusters. And these can give us a different perspective on our data, and we can have get an idea of the heterogeneity of our data sets. And this is quite effective when we have quite huge data set, and this could become a problem potentially. So as you can see here, we can you know, explore the cells and interactively shut off on the clusters to understand their similarity and their heterogeneity. Okay, so some of the last steps here that I will show you today. After we identify clusters, we might want to classify those clusters. So understand whether a cell cluster is cancer cells, T cells, B cells, or and so on. There are many methods to do that. Some of them are more automated. We can also approach these on a purely curation side. And often we do both. You know, I rarely rely just on manual curation or automated methods. I usually use both to gather more evidence. In this case, I just first show you how we can manually curate and understand the identity of this cell cluster. In this case, we are using a marker gene approach. And so what we are doing here is to use the function find markers to identify what marker genes define each of the cluster we have identified so far. And then we visualize the heat map of these markers. So we can evaluate how robust these markers are and basically visualize them. Now in our studio is a bit of a compressed view. This is a heat map function from Skater. But the idea is, as you can understand, that cells from the same cluster, they also cluster together by hierarchical clustering here. So that's a good sign, of course, not always the case. And here we have identified some markers toward the yellow color. And once we visualize this marker, we might want to research in the literature to try to understand what these markers are marker of, which cell type. And this is quite simple, is nothing particularly tidy about this, it's just using quite standard methods. In other methods, again, there are many. Here I will show you just one that automatically use reference signatures to classify our clusters. The reference signatures in this case is a Blueprint, and the package for automatic classification is called Singular. And so we simply gather Blueprint data and save and load it into a variable, and then we get some of our transcript abundance information, and we pipe it to a singular function. As you can see here, we are using Blueprint reference, where we specify the cluster identities of our cells, as you can see here. And this singular will output a table of cell identities. And this table, we will just convert it to a table, again, from easy tidyverse manipulation. And we select here the label, which is the cluster identity, and the cell type identity, as I will show you in a second. This might take a few seconds more. So again, this will produce a 2-column table, very easy. On one side, there is a cluster ID. On the other side, there is the cell type of each cluster. And I will show you in a minute, we can join, as we have done many times before, this new information to our single cell data and specify we want to join it by what, which is in this case, cluster ID. Sorry, he's hanging because he's waiting for, didn't notice. He's waiting for inputs. That's why it's taking so long. All right. Should finish very soon. As I mentioned, we have used Singular to detect for which cell cluster is the cell type is most likely to belong to. Okay, so we see natural killer, fibroblast, muscle cells, NK, so on and so forth. And as I mentioned, now we can join this information to our data set. Again, we are going back and forward from biconductor functionality and tidyverse. And in this case, that we have our cell type here. Okay, so pretty easy. This column has been added to our data set, as we have seen many times before. Okay, so we have another challenge. Again, a summarization exercise is quite simple. The question is, which cell type has the largest number of cells? So I've shown you how to identify which cluster has the biggest number of cells. this time we are summarizing on something else. And you can use Menti for this as well. And feel free to ask any question if you have now. How are we going, Maria?
Yeah, good. We've had a couple of people answer in Menti. So maybe give it another minute or two.
So just to specify, we have seen tidy summarized experiment for bulk data, and now we are seeing tidy single cell experiments. The difference that you might have noticed that for single cell data, by default, we interact with cell-wise information, not by default the transcript-wise information, although we can use joint transcript to add that information to our data sets. Now, just to show you, let's say maybe CD3G. There you go. Come. Okay, that's strange. Why? Let's see. Okay, sorry. Oops. Yeah, sorry. We have recently changed the name of this function. But as you can see here, the abundance of our feature, we have a new feature column and abundance column. And now we have our transcript information that we can use to do calculation, visualizations, and so on. On the other hand, because the data sets are smaller in size, for summarized experiment, we visualize everything in the abstraction. And as well, the other reason is that for single cell, the element we interact more with is the cell element, while is quite different from for bulk data.
Maybe just give another 15 seconds or so, yeah. And yeah. I'll end then this last challenge. Okay, I'll end it now. Okay, so which cell type first label has the largest number of cells? So it was a tie. So half the people said skeletal muscle, which is correct, and then half said B cells.
Good. Yeah, skeletal muscles, again, it's pretty easy in this case. We have used the count column count method before and arrange just to arrange them. As you can see here, actually, maybe we don't need, we just need first labels and we want to summarize by cell types. And so we can see, yeah, indeed, this is the answer. Congrats. This was the last challenge, but we will reveal later the winner of today's challenge. And I will show you the really last points of this tutorial. Now, I just want to mention that some of you might have already done or might be learning about pseudo-bulk analysis. Sometime we might want to simplify the data to do some exploration or statistics on the data. And so there is this concept of pseudo-bulk, where we might collapse the transcript information of all cells from one sample together to get a kind of bulk RNA sequencing data. Or, for example, we might want to group by samples and cell type to do some, bulk-like analysis. This can involve principal component analysis, differential expression analysis, and so on. This because there are a lot of tools that have been tested all over again and are very robust for bulk RNA data that can be applied to single cell RNA. In this case, we offer a function that is called aggregate cells, and it does exactly this. In this case, we ignore the cell type. We just want to collapse for each of our files that are roughly our data sets. And these, I often use pseudo-bulk analysis for different reasons. And in this case, is a good way to show that we can connect tidy single cell experiment with the tidy operation we were doing for bulk. And so we can apply now, I show you these objects. We went from single cell experiment to a summarized experiment. And you can see here abundance counts or log counts, our assays that we had in our object. And now we can apply the identification of abundant transcripts, the normalization, the scaling. And here we go. We can start with the analysis that Maria has just shown in a seamless way. Okay, so the last thing is I will briefly mention is basically the same as we already seen, how you can do this operation for Seurat objects. In this case, we have a similar Seurat object here. I can print it for you. As for single cell experiment, we get a rough summary of the object by default. But let's see what happened when we load tidy Seurat. As you can expect, now we get all the cell-wise information exposed to us as before that we can visualize and interact with. As again before, this is still a Seurat object, so we can apply our workflow as we were doing before with no difference. I apply here a Seurat function that is looking for the assays in our object and works no problem, of course. And we can do same operations as before for the Seurat. So the grammar of data manipulation and integration converged toward a tidy vocabulary for both single cell and Seurat. So there is a less cost for us to use Seurat rather than single cell if we want to switch, rather than learning a kind of area-specific, a package-specific vocabulary. We use the common vocabulary we know for Tidyverse. Okay, so this was it for single cell. You can have a read to the key points. So some basic steps of the single cell RNA sequencing analysis are dimensionality reduction, clustering, cell type classification. Single cell experiment, tidy single cell experiment and tidy Seurat is an invisible layer that operates on single cell experiment and Seurat objects and enable to visualize and manipulate data. Tidy single cell experiment is a single cell experiment object, as I mentioned many times. So nothing changed about what you were doing before. Nothing is taken away. We've just added this new bridge with the Tidyverse. So I will leave the closing remarks, and then we can have some questions and discussion to Maria. And yeah, I hope you enjoyed.
Well, thanks, Stefano, for that. Yeah, we hope you enjoyed the workshop and got something out of it. And to let you know, there is a solutions our markdown file that has all the solutions. If you can see it there in the files pane on the bottom right in Stefano's screen. And I have just updated the one that's in GitHub that creates the web page with the correct solution for that one that I showed in the first part that didn't work. It's just because we changed the data set, so the columns were a bit different names. So that should be fixed in the next half hour on the web page for the workshop. And other than that, we do have the little feedback survey that we would be very grateful if you could take a minute to complete and help us to improve future workshops, because we do take that information into account when we run future workshops. And also, yeah, we have time now for general questions and answers. So if you have any questions about bulk RNA sequencing, single cell, or these packages that we've been showing you, please do feel free to ask in the chat now, and we will address your questions. And just like to thank the Our Pharma conference organizers, Phil and his team, for the opportunity to give this workshop as part of the Our Pharma conference. So thanks.
Yeah.